Here today we are going to see about mutation. So mutation is the sudden change in the genetic material of an organism. So what is the genetic material? DNA. So if there is sudden change in the genetic material of the organism, we will call it as the mutation. And the term mutation was first introduced by Hugo de Vries in the year 1901. He observed some phenotypic changes in the evening primrose plant, which biological name is Enothera lamarckiana. So phenotypic changes means some changes in the outside appearance of this plant. And here the mutation is of two types. They are the chromosomal mutation and gene mutation. This gene mutation is also called as point mutation. So chromosomal mutation means the mutation will take place in the chromosomes, and gene mutation, here the mutation will take place in particular gene, that is the point mutation. This chromosomal mutation can be categorized into two types. They are the change in structure and also change in number of chromosome. So if there is change in structure of chromosome or the change in number of chromosomes. First we can see the changes in structure. So this change in structure of the chromosome may usually occur due to the errors in cell division. So this cell division is the one, when the cell division occur, then only our body will grow. The cells will divide and our body will starts to grow. So during the cell division, some changes will occur. So some deletion or addition of the nucleotides may happen in the chromosome. That is the changes in structure. We can see about that with one example. We all know that this is the structure of chromosome, and inside that the nitrogenous bases will be arranged, right? So just for your understanding I am drawing this A, T, G, C, A, and here also A, T, G, C, A. Like this the nitrogenous bases are arranged inside this chromosome. So if there is deletion is taking place, so this much space is getting deleted, this space is deleted from this chromosome. So how the chromosome will looks like? So this arm will be similar, but this arm it will be shorter than before. So because of deletion, the structure of the chromosome was changed. This is the change in structure. And next is the changes in the number of chromosomes. So this will also occur due to deletion, addition, duplication, inversion of the chromosomes. And if there is addition or deletion in the number of chromosome in a cell, that is called as the ploidy. So if there is addition or deletion, addition means adding, deletion means deleting, reducing in the number of chromosome in a cell, that is called as ploidy. And this ploidy is of two types. They are the euploidy and aneuploidy. First is the euploidy. It is a condition in which the individual bears more than the usual number of chromosomes. That is, we human have 46 chromosomes. That is 23 pairs of chromosomes, and these chromosomes are always arranged in pair. That is, it will arrange like this. Arranged like this. That is, in a single set, it contains how many chromosomes? 2 chromosomes in a single set. That's why it is called as diploid. Diploid is 2n. So each set contains 2 chromosomes. And the person who are with this euploid condition, they have extra chromosome in all these sets. So instead of 2, there it will present 3 chromosomes. And that condition we will call it as the triploid. Triploid, and it was represented as 3n. This is the triploid condition. And if the triploidy was found in plants and animals, they are sterile. Sterile means that plants and animals will not produce the offsprings or the young ones. But if it is tetraploidy, the tetraploidy plants, it was very advantage for that plant because it will produce more fruits, and also the flower size also will be very bigger size. Bigger size flower will be produced. This is the advantage of tetraploidy plants. So euploidy means increase in the number of chromosomes in whole set. Complete chromosome number will get increased. So here the triploid person will have totally 69 chromosomes. Normal person will have 46 chromosomes. For the triploid person they will have 69 chromosomes. That is euploidy. And next is the aneuploidy. So aneuploidy means here loss or gain of one or more chromosomes in a set. In a set there are 23 sets. In that 23 set, one or two sets. In one or two sets, the chromosome will get added or getting deleted. There it is of three types. They are monosomy, trisomy, nullisomy. So monosomy condition is 2n minus 1. So in a set, how many chromosomes will be present? 2 chromosomes, right? 2 chromosomes will present. That is 2n. 2n minus 1 is, in this 2, 1 chromosome will be get deleted. So how many chromosomes was present there? Only 1. Hence that stage is known as monosomy. Only one chromosome is present. Hence it is monosomy. Then next is the trisomy. In this trisomy, it is the condition of 2n plus 1. So now here two chromosomes are present in a single set. In that one chromosome is getting added. So now how many chromosomes are here? 3 chromosomes. So that condition is called the trisomy. Trisomy. Next is the nullisomy. Nullisomy means here it was represented as 2n minus 2. So we know that in a set 2 chromosomes will be present. So there minus 2 means these 2 chromosomes are getting deleted. Then how much will be there? 0. So that is null. Nullisomy. That is nullisomy. So here in aneuploidy, in monosomy 1 chromosome was getting deleted. That is it lost 1 chromosome. In trisomy it gained 1 chromosome, and in nullisomy it lost its 2 chromosomes. That is nullisomy. And the common aneuploidy disease is the Down syndrome. And this Down syndrome was identified by Dr. Langdon Down in the year 1866. That's why this syndrome got the name as Down's syndrome. And the people who have this syndrome, they will have one extra chromosome in the 21st pair of the chromosome. We know that there are 23 pair of chromosome. In this 23, the 21st pair have one more extra chromosome. That is 3 chromosome will be present in the 21st pair of this person. And this mostly this will be seen in children, and they have the symptoms like mental retardation, their mental growth will be poor, their development will be delay, they have problems in behavioral problems, and their muscle tone will be very weak, and they have problems in seeing and also hearing. These are the condition, and it will be seen mostly in children. And the next type of mutation is gene mutation or point mutation. This gene will be present inside the chromosomes, and these genes are made up of DNA, and the DNA is made up of nucleotides. So you can take this as the complete set of gene or the piece of DNA. In that the nitrogenous bases are arranged ATGC, ATGC, and in the complementary strand it was arranged as TACG, TACG, like that it was arranged. And if there is mutation means sometimes substitution will take place. Substitution means this G will always pair with C. So instead of this C, sometimes A will come and bind here, or T will come and bind here. That is the substitution. That is a mutation. So C only should bind with G. So instead of C, if any other nitrogenous base will come and bind, that is the substitution mutation. And the deletion is during the cell division, sometimes this pair will get deleted. That is this deletion. And insertion means sometimes one more pair will come and bind there. With this set, one more pair will come and bind there. That is the insertion. So like this all will happen inside this gene. That is the gene mutation. And we know that from this DNA only RNA will form, and from this RNA proteins will form. So the proteins which are formed from this mutated DNA will be abnormal. And because of that, one disease called sickle cell anemia was formed. And this sickle cell anemia, here the mutation will take place in the autosomal chromosome. So in the autosomal chromosome, one set of chromosome get affected, one set will get affected, and the person who have the sickle cell anemia, their proteins which are present in the hemoglobin will be in the shape of sickle. So the RBCs that carries the hemoglobin. RBCs only will carry hemoglobin, and this hemoglobin contain the protein, and that protein will be in the shape of sickle, and that sickle shaped hemoglobin will carry very less amount of oxygen, and so the person will be always anemic. Hence the name is sickle cell anemia. These are all about the mutation and its types.